Comparison of Cardamine hirsuta with Arabidopsis thaliana from research point of view. Cardamine hirsuta is a close relative of Arabidopsis thaliana, but it exhibits some morphologically divergent traits from its famous relative, such as in compound leaves, pod shattering, and altered root anatomy and trichome morphology. Become a major tool to uncover molecular mechanisms underlying the morphological variability. In Arabidopsis, the study of developmental mechanisms in compound leaf development, formation of multiple cortical cell layers, or explosive pod shattering cannot be performed in this species. Since then, the evolution of complex leaves from simple leaves of Cardamine hirsuta has proven to be an interesting model for several developmental processes, thereby making it a complementary developmental model next to Arabidopsis thaliana.
Where we will find these cardamine plants?
This cardamine plant can be found everywhere.
Yes, I seen this plant in Monar Hills.
And also I went to buy some ornamental plants. Also found some cardamom plants are growing in that pot.
How do you identify this as cardamine plant?
And itself giving a hint. It is a cousin of mustard plant and it is a small plant. I attracted by seeing the seed pods and it having beautiful white flowers.
Such a heavy terms.
A simple way. This plant is having flowers, so this is flowering plant. And when this is at juvenile stage, it has two leaves named dicotyledons. And this belongs to Brassicaceae family.
Wow, it's a kitchen family.
Flower is similar to all others flower as it contains sepals and petals which are 4 in number or tetramerous. Male and female reproductive part is found in a single flower called bisexual.
The arrangement of stamens is tetradynamous. That means 4 plus 2 arrangement. 4 inner long stamens and 2 short outer stamen. Cruciform corolla is the important character of family Brassicaceae, in which the free petals are arranged in the form of a cross.
It is not a single flower. Flowers are seen in cluster form. Actually, it is a raceme, but it looks like corimbos. In the initial stage, all the flowers reaches at the same level. Flower mature and pod will form. Due to elongation of pedicel, the oldest flower is at the base and young root is dry and seed pod will burst out the seed when it reaches to the maturity.
We can see different types of species around us, but the commonly available cardamine species are Cardamine hirsuta and flexosa. So, we are in the process of identification.
Rosette leaf of arrangement is seen at the base of Cardamine hirsuta. Basal leaves are hairy. Leaves which are present on the erect stem are colline, which are minimum in number. Stamen 4. And seed pod are present parallel to the axis of stem. In cardamine, two types of leaves are present. Basal leaves with entire margin, compound leaves with distinct leaflets.
The life cycle of cardamine is different in different places. Sometimes it may because of species difference. In Ranji, from seed germination to next seed formation, it will take 60 to 65 days. In Kerala, it is observed that 40 to 45 days.
Cardamine plants can also be used to study genetic engineering by floral dip method. Why floral dip? Why not any other conventional method? Well, the floral dip method requires considerably less time, effort, and results in greater seed yields. It is simple procedure for transformation from the other conventional method such as tissue culture, which requires time, skilled labor, and relatively expensive laboratory facilities.
The entire process starts from growing healthy plants until it starts producing the floral bud or inflorescence. Next, when it comes to that stage, start culturing the Agrobacterium strain that will harbor the gene of interest. Then, resuspend the cell in a freshly made 5% sucrose and silvate solution. Then invert the plant and dip only the aerial parts of the plant in the Agrobacterium cell suspension for 10 seconds with a gentle agitation. Then cover the dipped plant with a plastic cover or wrap them with a plastic film and lay down the treated plant on their sides for 16 to 24 hours to maintain the high humidity. Allow them to grow in isolation and also collect the dry seeds. Check for the transformation. In this case, we used the kanamycin resistant to check for the resistance.
How will you determine germination rate of transformed seeds? For that, I need to determine germination rate of non-transformed seeds. But due to lockdown, I don't have non-transformed seeds at my place. Oh, I found some cardamine plants at my place. Let me help you out there for determining germination rate of non-transformed seeds. But how could you do this experiment at your home during this lockdown? Why not at home? What exactly things are needed for germination? A container, a water, and of course cardamine seeds. Let's try this out. For this, I will need kanamycin antibiotic at different concentration, tap water, and non-transformed seeds. How are you preparing different concentration of kanamycin? For that, first I will be preparing stock solution at different concentrations such as 10%, 30%, 50%, 70%, and 90%, and then serially diluting it. 20 seeds in control and 20 seeds for each concentration followed by 2 replicates. Whoa, I am very excited for your results. Look what I found is kanamycin is not affecting germination rate as 80 to 85% of germination is observed, but it is affecting the plant growth. How? Two factors has been observed. Etiolation and hypocotyl reduction is observed as concentration of kanamycin solution is increased.
That's great result, but at what concentration are we observing the visible effects of kanamycin in non-transformed seeds? Well, in 50 mg per liter, we can see the visible effect of kanamycin in the non-transformed. But I also need to find the rate of germination and growth of cardamine seedling in the presence of kanamycin solution. Well, don't worry, let me help you out in this part too. But why are you only choosing kanamycin antibiotic? Why not others such as penicillin?
Antibiotic resistance in plant. Why not use penicillin? Penicillin kills bacteria by affecting its cell wall. But as we have studied, cell wall of bacteria is different from plant cell wall. Kanamycin on the other hand affects the ribosome of bacteria, and cell organelles of plant cell like chloroplast and mitochondria have bacterial origin. That is, it contains bacterial ribosome. So theoretically, kanamycin should affect plant cell. Let's find out.